Ko taunaki toku monu. Ko watara toku awa. Ko tiati awa toku iwi. Ko ngati wahiri toku hapu. Ko owai toku morai. Ko Richard Faull toku ingna. Ngā mihi kia koutou, tēnā koutou, tēnā koutou, tēnā koutou katoa. It is wonderful to be home. Great to be here. Meet so many of you who got connections either directly with me or through my mum and dad. And of course, as you all know, I'm really, we'll advance this a bit, I'm really a Taranaki boy come home. And that's a privilege which is fantastic. And one of the themes I'm going to be tonight is going to be, the reason why I'm here is to give you a bit of exciting news about the research we're doing. But most of all to say thank you, because you people have enabled us to do what we are doing. And I'll explain that as we go through. And it's, the fact that I grew up in Tikarangi under the shadow and the guidance of Mount Taranaki. And, if you look carefully here, you can actually see Tikarangi. And that's where I grew up. And here's a close-up, you see. Many of you who I met tonight, part of the pioneer families that went Tikarangi with us, with my mum and dad. And of course, this is the 5 boys and my dad. And since they're not here, I can say I'm the best looking one right in the middle there. And my dad, through him and through our Granny Rolf, that we have this proud whakapapa to Ngāti Rehiri Te Ātiawa. And that is special. Also, of course, we whakapapa back to our ancestors, from Cornwall, from England. It's what I call having a foot in both camps, and that's got to be good. And, of course, we grew up, and my mum and dad had the general store. And the general store was the best environment to grow up with, because my mum and dad, they were wonderful parents, and they gave us values. And the values that they gave us are actually the values which enabled us to succeed in research. So they taught us first, that's my mum and dad there. Many of you spoke to them when I met you when you came in, and you recognise them. They were pretty young in those years. And so one thing they told us is that you look after the people. All of us 5 boys, we deliver the groceries. And my dad said, we will do anything to help anyone. And that's the best message I have. The best I never realised it till much later, that the things we learnt back home, in Taranaki, in Tikarangi, in the shop, were the ingredients of really being humble, making sure you go the extra mile for someone else. Don't think about yourself, but be true to yourself and look after other people. Be honest, be humble. And my mum taught us to be passionate about life and never give up from what you want to do. And just do your very best and try and be the best in the world. But above all, what they told us was show dignity and love to all people. And just look after the people. And of course, my Māori whakapapa teaches us that. He tangata, he tangata, he tangata. I never realised it in those days. But that's what life is about. No matter what your culture is, that is what life is about. So tonight I'm going to take you on a journey. It's going to be the journey of my life in brain research, a journey which you people here, you don't realise that you made me take that journey. And many of you here, many of the families here tonight, and throughout the whole of Aotearoa, New Zealand, they supported us by actually giving the brain of their mum or dad after death who had a neurological disease for us to do research on. So I'm going to tell you that story and tell you first tell you a little bit about the human brain itself and what got me captured on it, and then tell you about how we formed the Human Brain Bank, And with the wonderful support we've got from the Neurological Foundation, you know, they're celebrating their 50th anniversary, which is fantastic. And you people, the people of Aotearoa New Zealand, have funded the Neurological Foundation to help this happen. Now, it's a sheer coincidence that the Neurological Foundation is celebrating 50 years, and it's 50 years since I started my PhD on brain research. I'm not saying there's any equivalence here, but here we are. I look too young, I know. So, here we go. So that's the human brain. I saw it as a young third-year medical student. I'd just gone down to Otago Medical School, and I didn't know anything about the human brain. I saw it as a young third-year medical student when we started looking at the brain. And every Saturday morning we would sit down. Yeah, we actually went to classes on Saturday morning those days. They were great days. And we carefully dissected it, pulled it apart. And I could not believe, a little Taranaki guy, I could not believe how this human brain is what makes every one of you here tonight special. It's what you are. You are the human brain. Your brain is you. Your passion, your personality, all your idiosyncrasies, all your qualities, why everyone here is special, why you've all done certain things in life. And they're world leading, excepting it doesn't necessarily get on the front page of the paper, it doesn't have to. But you're all special, and it's because of that wonderful human brain. Now, the human brain is quite unique, and this little rat brain here, see, this is a monkey brain. And you see the rat brain's got the smooth outside of the cortex here, and then as you go up through the animals, it's that part of the brain which starts to get more complicated. In the human brain, we are the pinnacle of brain function. And that has become so expanded, that outer cortex, into all these different folds, that we are the most complicated, it's the most complex organ in our human body. But it's what makes us who we are. And that's what intrigued me, you see. And you see all those little folds, they're quite regular. There's actually a pattern to it. No matter whether you're male or female or whatever, you have the same pattern. You can look at a brain, you can't tell whether it's from a black person, a white person or whatever. You can't tell whether it's man or woman. They all look the same on the outside. It's on the inside that makes you different, you see. And so we have this fold that comes down here, and we're looking at the left half of the brain, the same as on the right half. This controls all your movement, all your muscles moving on the opposite side. Here, just behind it is a fold that controls your sensation. It enables you to consciously feel every little touch on your skin, every kiss on your lips. You don't feel it on your lips, you actually feel it right there. That's where you feel it, because if that's affected by a tumour, your lips are numb and you won't feel it. And then we have the area at the back for vision, here for listening. And what's got huge in the human brain is the memory area. But what makes us so unique to all other animals, that we have a complicated area for reading and writing and for speaking. Now, humans would not have advanced in civilization if we couldn't read and write. If we didn't write it down and learn, every generation, every minute of every day, we advance knowledge. Rats and cats and monkeys are doing the same today as what they're doing 1,000 years ago. Today, life is changing. It's changing fast. And it's because of our cerebral cortex, right? It's because of your brain. Some people's brains work wonderfully. Some people get a few screwed up ideas. You know, people like Trump and all the rest. Anyway, I won't go there. A part of the brain in the human which is massively expanded is this area at the front, what we call the prefrontal cortex. That's where your personality lies. That's where your executive planning lies. That's where your vision for the future lies. All that is ingrained. All that is developed. When you get an idea, you wake up in the morning, you still know who you are, and you know, this is what I'm going to do today. This is what I like to do, and this is what I'm going to do for my life. All of that is in there. And you take it for granted. I guarantee no one in this room, when they woke up this morning, said, "Thank you, brain, for making me who I am," right? But you should do that. It's the most priceless asset you'll ever have. And it's absolutely mind-boggling. Just talking to you now gets me excited, you see. And, But of course, we're just looking at the outside of the brain. Inside the brain, there are billions of brain cells. Each individual cell here, you see, they're what we call neurons. 60, 100. We think there's about 100 billion brain cells in each of our brains, in general terms. Now, how many is 100 billion? Well, there's 6 billion people on Earth. Imagine that if everyone on Earth was a brain cell, then it's at least 10 times the number of people on Earth inside your brain. And they're talking to each other, you see. But they're talking to each other in the most coordinated way. Each single brain cell gets input from about 120,000 other brain cells. And it integrates and synthesizes that information, gives you wild thoughts, gives you happy thoughts, gives you sad thoughts, gives you a vision for the future. And if that's not magic, I don't know what is. And that is the human brain. And that's human brain 101. I told I can't talk past midnight tonight, all right? So I've got to keep going, unfortunately. But isn't that wonderful? And it doesn't matter who you are, we've all got, on average, 70 kilogram person's got a 1.5 kilogram brain. 20 grams of brain weight per kilogram of body mass. With your male or female, it's the same. It just so happens that most males are heavier than females, and so they have a bigger brain, but it doesn't mean anything. So you see, it's I get paid for what I love doing, and that's got to be special. And so I started out as a young medical student. After I saw the brain in third year, I thought, God, I took a year out and did brain research. And of course, you can't do it on the human brain. You can't do experiments on people. You have to do it on the rat. So I started on the rat. Wonderful. It was a scaled-down model of the human brain, but it's still fascinating because you can learn a lot of things to see about neurological disease. And then I really wanted to keep going doing a PhD, but my professor of anatomy said, no, you finish medicine, and then you come back and start a career in brain research. But finish training as a doctor, because that will give you a link to clinical medicine, which is actually critical. It will keep you humble. So, finished medicine, tried neurosurgery, loved neurosurgery, but suddenly realised we didn't know very much about the brain. And so I went back into the PhD and spent 4 years in Auckland University doing a PhD, and found a new pathway. Is this in the rat brain still? Pathway about how we control our muscles. Then went off to the United States for 3.5 years, Diana myself, and had a most wonderful time. 1 year in California, working at the NASA Ames Research Centre. They were seeing what happens to the brain when they go into space, and learned a lot from a great person there, Bill Mailer, world leader in brain research. Then went off to Boston and studied at MIT and Harvard, 3.5 years there. Professor Wally now, the world leader in brain research. Learned new techniques, came back to Auckland, 1978, and I started my own little lab at the Auckland Medical School, quite a small medical school at that stage, and started working on the rat brain, looking at the parts of the brain which control movement, which are affected in Parkinson's and Huntington's disease. And then something happened in 1980 which changed my life forever. Professor Arthur Veal, actually his father was Paul Veal, he was my chemistry teacher at Newport Boys High School. He was the expert, New Zealand expert in genetics, and he said, Richard, I look after all the people in New Zealand, in Aotearoa, New Zealand, who have Huntington's disease, a tragic disease which is caused by a gene. It's quite a rare disease, thank goodness, but it's caused by a gene. It's a dominant gene, so if a mum or dad have it, the children have a 50% chance of getting it. We've got a number of families here, whānau here, who are Huntington's whānau, who we're working with to try and help them. I'm seeing them tomorrow, and we've got good news for them. Actually, I'm going to tell them tomorrow. I won't tell you tonight. And so he said, we don't know the gene that causes this disease. We know it's caused by a gene, but the families, we can't test for it. You see, the families want to know, do they really have this disease that can be confused with Parkinson's disease? And so he said they would really like to know if this disease is really in their family, because if it is, the implications are huge, you see. Would you look at the brain of their mum and dad when they die so that you can tell the children whether it's yes or no? And what he asked me to do, you see, this is a section from a normal brain. This is from a person who died with advanced Huntington's disease. And you can see the marked atrophy here in what we call the basal ganglia. And so he was asking, he said, can you look inside the brain after death? The family will give the brain to you, and if you can give them the answer. And I said, sure, absolutely, in any way we can. So that's what we did, you see. And the most amazing thing is that because he looked after all the Huntingtons families in Aotearoa, New Zealand, he started bringing us brains from all over New Zealand. He would bring it in a plastic container, fixed in form. This is something from the family. Look at it and tell us what the outcome is. And so we would look at it, teamed up with the neuropathologist at Auckland City Hospital, and we would look at it, examine it, do special stains on it. Then we would call the family and talk to them. And we'd say yes or no. In most cases it was yes, but in some cases it was no. And that was transformational. But what was transformational for me was that these families then said, well, you keep mum's brain, dad's brain, and you do research on it. Find out why those cells are dying so that you can help our children with new research. And I said, that would be an absolute privilege. And that started me off. The family's given me the brain of their mum and dad, told me get to work in the lab, do research and find out what's happening here. And that started my whole career on the human brain research. And we have family here tonight, Whānau tonight here, who've done just exactly that. So that's why I'm here to say thank you for giving our research team, giving neurological research in New Zealand through the Neurological Foundation Brain Bank, the brain of your loved one, and entrusted it to us. We're the custodians so that we can crack and do world-leading research on the human brain. Okay, so what we were establishing, see, was a bit of a partnership between brain scientists and patients and families, but really families were driving it, you see. That's the fascinating thing about it. They were saying, now get going, help us. And when we used this tissue, we found that we could do all sorts of studies on it, genetics, chemistry, anatomy, pathology, so we involved lots of different brain science teams. We went to the doctors who looked after that person during life and got the whole clinical history. We went back to the families to get more information. And we were building up a network, you see. We were communicating in a network. And I say this network, you see, is sort of it's the triad of brain research. The Holy Trinity, if you like, of brain research. And we found unexpected findings. And it's helped us to find new treatments, which I'm going to tell you about towards the end of my talk. So, and in 1994, we gradually started collecting brains, not just Huntington's brains, but Parkinson's, Alzheimer's. And we started, the first brain we got in 1981, and we'd already started collecting other brains. And so I went to Neurological and I said, would you help me? We need to get money to make this into a really efficient human brain bank so that we can do better research. And so as the years went on, we extended into other diseases. The most recent one is concussion and sports injuries. You've heard about it on the radio recently. The tragedy that playing sport, which is supposed to keep you fit, can sometimes be detrimental to your brain if you keep getting concussion and knocks. So we're going to do research on that under the Neurological Foundation Human Brain Bank. And this keeps us helping people, you see. And we get support from all the different organisations. The Neurological Foundation give us this wonderful funding. We got 4 staff, fully funded, money to analyse it now. We started off, and we had to fund it from our own little research grants. But thank you, Rich. Thank you for the foundation for enabling us to do what we can do. And also, you see, the tissue is available for other research groups, not only Auckland University, our Centre for Brain Research, but also other people in New Zealand and overseas. And this is what a world-class human brain bank looks like, you see. Well, this is just one room. This is where we actually do the section of the brain. This is some of our critical staff. And we carefully, over the years, we've developed a method of doing this so that we pull the brain apart, if you like, into little blocks. It's like taking a Lego. You pull all the different blocks apart, you give each of them a code, and we know exactly where each one comes from. About 60 blocks on one side of the brain and 60 blocks on the other side of the brain. It's about 120 blocks per brain. So we've got tissue from over 1,000 brains, about 45,000 at least blocks of tissue. And that is the magic of doing successful research on the human brain. I would like to stand here tonight as a man from Taranaki saying it was my idea, but it wasn't. It was the people's idea. It was the family's idea. That's why when my mum and dad said, go out and help the people, I didn't realise it till 30, 40 years later that the message they gave me then was making my research successful. And so that's why I'm here tonight, to thank you. So when I say thank you, you didn't realise I was going to thank you. But that's why it's lovely to have so many people here tonight, to have this occasion to celebrate the 50th anniversary of Neurological Foundation, and to do it in a way that comes from the heart. And, you know, my success actually is your success. Our team is teamwork, nothing more than that. And when I used to, you know, every paper, when I ever wrote a research paper, I'd send it home to my mum, a copy for her to read. And she'd read it. She said, well, I don't understand much of this. She said, but just get on with it, she said. Just get on with it. And that's what I've been doing. And so you see, because we knew the history of every single brain we had, because we could ring up the family and ask them and get the details of that history, we had the most wonderful, the most precious brain tissue, human brain tissue in the world. They've got a brain bank in New York, they've got one in Boston, they've got one in Cambridge in England, they've got one in Zurich, but they don't have the connections in the family which you can do in a small country like New Zealand, you see. So Aotearoa New Zealand is unique. You know, Kiwis can do anything, and we can even do human brain research better than anyone else. And it's because of the family. So we send tissue with the family's consent to all the top groups around the world to do brain research, because they can do things on it, they've got more money than us, they can do certain things that we can't do. But they're using the tissue from our Aotearoa New Zealand donors. When we ask the family, we ask every family, are you happy for us to send a little bit of mum's brain to Zurich? Dad's brain to Oxford or whatever? Every single family has said, yeah, go for it. And we get all sorts of comments coming back. Some of them, they say, oh, it's like mum's getting the travel and death she didn't have in life. Or dad's going to live on forever through the human brain bank, looking after his children through research. And that makes you humble, and it makes us get on and say, let's push this along. So this sort of triad of neuroscientists working together, collaborating, working as a team, working with families, working with doctors in the hospital, is something pretty unique. And across Auckland University, you see, we have developed a club, a special club. All these people, all the brain scientists at Auckland University belong to a club, and we called it the Centre for Brain Research. We launched that in 2009, and I call it, some of us call it the All Blacks of Brain Research, but it's just teamwork, you see. And it's great working as a team, because you get all these new ideas. The families give you ideas. I'll go back and talk, and we always go out and talk to families. I tell all my team, go out and talk to the people who have the disease which you're doing research on, and tell them what you're doing. Give them hope. They want to know. They say, oh, yes, but we don't have a cure. I said, they'd love to have a cure, but they would just be turned on by the fact they know you're doing research, you see. It gives them hope for the future. And also, they ask you questions. Well, I'm going to get questions here tonight. Questions which give us new ideas. Because we're right in the middle of it. And sometimes you need someone outside saying, hey, what about this? What about that? And that's why we formed the Centre for Brain Research. Yeah, so this is the Centre for Brain Research. You see, we've got 3 pillars We've got the researchers at the university, we've got the doctors in the hospital, but most importantly, we have the community, the families affected by brain disease. And that's what it's all about. So I'm going to tell you, in the brief time we've got left, I'm going to just take 3 examples of our research and just give you a little bit of a teaser about how we're changing the world, of our understanding of the human brain, how we're changing the world and how to develop new brain disease. I'd love to go through everything we've done, but I've just got 3 examples, something which will turn you on, and you'll go away tonight and you'll be different people. You will not be the same after I've heard what I'm going to tell you, right? And if you don't believe me, just ask for a refund at the door, okay? So, the first thing we found, and we published this in 2007, we're doing more work on it now, we discovered that the human brain can make new brain cells. Now, when I was brought up, I was told, look after your brain, because you're not going to be able to make any more human brain cells. You know, once you got to maturity, that's it for life. But we actually showed that you make new brain cells throughout the whole of your life. You may make fewer as you get older, but you make them forever. So everyone here tonight, me included, it keeps me going, you see. I come out and talk to you, it gets me excited, it gets me new ideas, I make new brain cells. But most importantly, we showed the pathway which those new brain cells take through the human brain all the way down here to the smell area of the brain. But this is the motorway of new brain cells. It starts right in the middle of your brain. And we published that in the top journal of Science. There's a big story there which I haven't got time to tell you about. But initially, no one believed us. And we had to go back and do more research and send it off to, we first tried Nature. And they said, no, we just don't believe this, you know, little Kiwi scientists, you know, way down the other side of the world. This is, you know, which we've looked for this path, but we weren't able to find it. But we found it, you see. So we had to put more results together and send it off to Science in the United States. And after a bit of to and froing with some referees who they realised this was actually a total breakthrough. This changed our thinking on the human brain forever. This means that throughout the whole of your life, you can actually make new brain cells. It was revolutionary. It was revolutionary almost as Galileo in saying that the sun doesn't go around the Earth, but the Earth goes around the sun. It was way back. So, and this was controversial. It still is controversial. Some people didn't believe us initially, but it's been repeated, and we were right. The most interesting thing, because we know we make new brain cells as you know, in the human brain, then all our studies on animal brains, where we know they make new brain cells, and we know that studies in rats show that if you put rats into a stimulating and enhanced environment, give them lots of things to do, running wheels to play with, get their brain switched on, they will make 40% more brain cells. Give them exercise, make more brain cells. So we can't do this experiment on humans, but we can extrapolate those results down, you see. So if I've turned your brain on tonight, you've made a few new brain cells. And if you exercise, don't stress your exercise because you'll make less. Just do it in moderation. Keep fit. Keeping your heart healthy keeps your brain healthy and keeps making new brain cells. So that was incredible. So that was something totally new. You see, we can only do that because the brains were given to us from whānau, from family, who said, find out new things about the human brain. And that was one of them. I like to think that creative thinking and stimulating thinking makes new brain cells. You know, no matter what you do in life, you think creatively, in a creative way, new ideas, do this, do that. That's actually turning your brain on. You feel better when you do that. The more you socialise and interact with other people, we know that can turn your brain on. You'll be making new brain cells. So, use it or lose it. That's the key. And now we're doing much more work now, finding out whether there are motorways coming off this big central neurogenesis pathway to see if it goes off into different parts of the brain, Huntingtons and Parkinson's and Alzheimer's and stroke. So we've got our work cut out for us. There's a lot to do. And it's pretty exciting stuff. Gets me up in the morning, gets me going to work. Well, it's not work, it's just going to have fun, you know. Expand the horizons of the human brain, and that's got to be incredible. Second example. Because we work closely with the neurosurgeons in Auckland City Hospital, it's the biggest neurosurgical unit in Australasia, because they take out pieces of brain tissue to help people who've got a brain tumour or brain disease, we get permission from the families saying, can we take a little biopsy of that tissue that's been taken out? Instead of it all going to the pathologist, how about giving some across the road for our Centre for Brain Research? And then we take the bit of tissue and we carefully look after it, cut it up into small pieces, dice it up, create cultures of those individual cells, keep them alive. These are just individual cells. We're not keeping your brain alive, just individual cells. Keep them alive in the lab and do experiments and do analysis and see if we can find out new drugs to help the disease which the person who's given the tissue to us, who's still alive, but had the tumour removed. And so you see, we can culture human brain cells. If I'd said this 10 years ago, I'd be fired. And this is what healthy human brain cells look like, just separated and kept in a dish in the lab. And this is what cells from a person with Parkinson's disease. They're less healthy, you see. So this is a disease in a dish. We trial drugs. We're doing a lot of work on cancer of the brain. And we've got a few leading possibilities of some particular chemicals which will slow down those cells from multiplying and forming a big cancer. And we're going to be doing drug trials on that. It's exciting. That's giving people hope, you see. So that's the second example. Third example, it's all about Huntington's disease. And looking at the human brain from families, the brain given to us with their mum or dad who died of Huntington's, we've been able to understand what's going on inside the human brain, working with people overseas. And we understand what's going on with the human brain. We know that's caused by a gene. I'll show you the gene in a moment. And so there's been a lot of work using rats and putting a small part of the gene into rats and seeing if we can work out what goes on, what does that gene or part of the gene, how does it affect the rat, and how does it make proteins which end up causing Huntington's disease. Those results have not been very successful. So it was our idea to actually put the human gene for Huntington's disease into the sheep, because the sheep brain first is much more similar to the human brain. And being in Aotearoa, New Zealand, you sheep. And so you see the human gene lies right at the tip. We all have a gene we call IT15 on our chromosomes. This is all the chromosomes. Human chromosome, the 23 pairs, X and Y is there. And we have 2 of each chromosome, of course, one from your mum, one from your dad. And if you get there, and we've got a gene up here called IT15. And when that gene gets particularly long, it turns into the Huntington's gene and will cause this tragic disease. We now know about that. It was actually cloned and identified in 1993. So we took this gene, we actually got it from colleagues at Harvard University with the family consent, and they had isolated the gene, and we took that gene. It's a big gene, huge gene. That's why in rats they only put a little wee part of the gene into the rat brain. In the sheep brain, we thought bigger is better, so we went to put the whole gene into the sheep to see if we could mimic the results in the human and then see if we could turn that off. And so in a very intricate process, which using top-class geneticists in both in Aotearoa, New Zealand and in Australia working together, we were able to actually get fertilised eggs from the sheep, take those eggs into the lab, put the human gene, inject it into the eggs, fertilised egg, then reinsert that back into the ewe, and then create a whole series of animals which had the human Huntington's gene in it. This took about 10 years to do that. And, the Freemasons of New Zealand actually gave us the money to do this because no one believed we could do it because it's such a big gene, but we did it. And so what we've got now, and I'm just cutting this way down short now, we've got a whole line of sheep in Australia. We actually created 6 different lines. They're on a big research development farm in just north of Adelaide called the South Australian Research Development Institute. It's a big government institute and specially bred to make what we call transgenic sheep. Sheep with the gene of a particular disease, in our case, Huntington's disease. And we've got 6 lines established, which look very promising. Out of those 6 lines, we gave them all a name, you see, as we were developing them. The best line turned out to be the Kiwi line. The Aussie line turned out to be a runt, actually. So anyway, so now what are we doing with this? We are using this to develop a technique where we can inject chemicals into the brain that will turn that gene off. And this is something we never dreamt we could do. We are doing this in association with international collaborators in England, in Switzerland, and especially in the United States, Boston. Our colleagues have been with us, who we have given human brain tissue to us, and they're helping us now to crack the code, you see. And within the next 5 years, I'm going to tell the Huntingtons whānau tomorrow, when I see them, we're going to be able to give a particular, specific, what we call oligosense, special gene suppressor to turn that gene off. And that will be the most wonderful thing. When I first started off with Huntington's disease, a euphemistic said, we're going to try and get a cure for this disease. God knows where it's going to come from. But you know, we're almost there. And that's the most satisfying thing ever. So you see a little Taranaki boy growing up in Tikarangi, mum and dad in the family shop. You learn things which carry you on forever. And as they said, never give up on your dream. Go for it. If someone says you can't do it, say you can. Be imaginative. Always go for the top of the mountain. You may only get halfway up, but you'll get a lot further if you aim for the top than if you actually aim for halfway up. And that's the story of our brain research to date in Aotearoa, New Zealand. And so the Neurological Foundation Human Brain Map, you see, has been at the centre. But you know, with due respect to the wonderful funding Neurological Foundation give, the people who are at the centre are the people. The families. The Neurological Foundation exists to help people. We exist so that the Neurological Foundation can provide us with support so that we can do this research. I would never have thought going to Tikarangi School, then going to Waitoa High School, I could never have dreamt an exciting dream as much as this, you see. So I tell my graduate students, I say, dream big. Don't worry about the minutiae. Worry about the big picture. And make your dreams come true. Coming here tonight and talking to you and just saying, giving you that message, you see. You all make dreams come true in all your lives. And New Zealanders and Kiwis, we have this, we are innovative. We have new ideas. We can beat anyone with less resources. And brain research is no different. By growing up in the right environment, given the right messages, given the right values, and you can turn even failure into success. I always say, there's no such thing as failure, it's success in disguise, because if something fails, you just turn and try it a different way and get on with it. So thank you all for coming tonight. Cold night, had problems parking, coming in here and meeting many of you who knew my mum and dad, who I've known, who I've grown up with, relatives here tonight, and everyone. It was a very humbling experience. It was wonderful to come back and just tell you a little bit about what's going on in our world, and that's going to help your world, you see. Thank you very much.